Part 2, the oxygen-hemoglobin dissociation curve. In this tutorial, we'll briefly discuss the binding between hemoglobin and oxygen, and how this binding is represented on an oxygen-hemoglobin dissociation curve. From this curve, you should be able to determine a value known as the P50. In addition, you should be able to predict the effects of different physiologic conditions on the binding between hemoglobin and oxygen, and therefore the effects on the dissociation curve and the P50. And we'll also compare oxygen binding by normal adult hemoglobin with fetal hemoglobin and with myoglobin. The cytoplasm of red blood cells is filled with hemoglobin, and this pigment is responsible for the red color of these cells. Hemoglobin is specialized for oxygen transport, and about 98% of the oxygen transported in the blood is bound to hemoglobin. In this lab, we'll focus on the properties of binding between hemoglobin and oxygen. In the lungs, oxygen diffuses into the red blood cells and binds to hemoglobin inside the red blood cells. The red blood cells are then transported to the tissue via the blood. And in the tissue, the hemoglobin releases the oxygen to meet the metabolic needs of the cells in that tissue. Let's briefly review the structure of hemoglobin. Structurally, hemoglobin is composed of 4 polypeptide subunits, 2 identical alpha chains and 2 identical beta chains. And each chain is conjugated with a non-protein prosthetic group called heme that you can see on the right side of this figure. Heme consists of a porphyrin ring structure, and at the center of the ring is a ferrous iron ion, right here, that's capable of reversibly binding to one oxygen molecule. So an oxygen molecule will bind right here. So there's 1 oxygen molecule per heme, and 4 heme groups per hemoglobin molecule, and therefore each hemoglobin as a whole is capable of transporting 4 oxygen molecules. So the hemoglobin molecule can bind oxygen here, 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 and here, 1 on each subunit. And when the hemoglobin molecule is completely loaded with 4 oxygen molecules, we say that it is saturated. So you can see on the right side of this figure that the hemoglobin is fully saturated with 4 oxygen molecules. And physiologically, you'll most likely see saturated hemoglobin in the lungs, where the concentration of oxygen is the highest in the body. A hemoglobin molecule that's bound to 1 or more oxygen molecules is called oxyhemoglobin. And we sometimes see this written as HbO2. Now, the red blood cell that contains this oxyhemoglobin molecule will travel via the blood to the tissues where it's going to release or unload the oxygen. Now, the red blood cell that contains this oxyhemoglobin is going to travel via the blood to the tissues where it's going to unload the oxygen so that the cells that require the oxygen can use it. But the oxygen is not unloaded all at once. Rather, 1 oxygen dissociates from 1 heme, and then the next oxygen dissociates, and so forth, so that a hemoglobin molecule may be present in various states of desaturation. When the hemoglobin molecule is completely desaturated, we call that deoxyhemoglobin. And that's usually just written as hemoglobin or sometimes HHB. Now, it's not very common to have completely deoxygenated hemoglobin. At any given concentration of oxygen, a portion of the hemoglobin is typically in the form of oxyhemoglobin. And you can demonstrate the percentage of oxyhemoglobin by plotting an oxygen-hemoglobin dissociation curve. And this slide shows a typical oxygen-hemoglobin dissociation curve. And you may also see this curve referred to as a hemoglobin-oxygen dissociation curve, or an oxyhemoglobin dissociation curve, or simply an oxygen dissociation curve. And this graph plots the percentage of oxygenated heme groups as a function of the partial pressure of oxygen in a tissue. The partial pressure of oxygen is a measure of the concentration of oxygen in the blood or tissue fluid, and the units we use for partial pressure are usually millimeters of mercury. And the y-axis is the percentage of heme groups that are oxygenated, and you also might see this designated as the percent hemoglobin saturation, or the percent saturation of hemoglobin. What this means is when the hemoglobin in the blood is 100% saturated, every hemoglobin molecule carries 4 oxygen molecules. And if the hemoglobin is 75% saturated, then there's an average of 3 oxygen molecules bound per hemoglobin. And if it's 50% saturated, then there's an average of 2 oxygen molecules bound, and so on. One important feature of this curve is its shape, and that shape tells you something about the dynamics of the binding between oxygen and hemoglobin. Take a moment and think about how you would describe the shape of this curve. The shape of the curve is what we call sigmoidal, or sigmoid, and that's sort of shaped a little bit like an S. And we'll talk about the significance of that shape in the next slide. The sigmoidal shape of the oxygen-hemoglobin dissociation curve is typical for multi-subunit molecules that exhibit what we call cooperative binding. Cooperative binding means that if you start with an unloaded hemoglobin molecule, when the first oxygen molecule binds to any one subunit of the hemoglobin, this is going to induce conformational changes in the hemoglobin such that the remaining subunits have greater affinity to bind the next oxygen molecule. So the affinity of hemoglobin for oxygen increases as successive oxygen molecules bind to the hemoglobin, and this cooperative binding is reflected in the shape of the curve. As the partial pressure of oxygen rises, there's a rapid increase in oxygen loading onto hemoglobin, and you can see that in this portion of the curve here. Ultimately, when the partial pressure of oxygen is high enough, oxygen is bound to virtually all of the binding sites on the hemoglobin, and at this point, the curve plateaus, as you can see here, in this portion of the curve. And here, hemoglobin approaches 100% saturation. So even if you increase the partial pressure of oxygen, there's not much further increase in binding to hemoglobin. So the take-home point of this slide is that the sigmoidal shaped curve is indicative of cooperative binding. And keep in mind that cooperative binding is only possible for molecules like hemoglobin that have multiple binding sites. Another important feature of the curve is the P50, which is also called the half saturation pressure. And the P50 is defined as the partial pressure of oxygen at which 50% of the hemoglobin is bound to oxygen. And you can also say it's the partial pressure where hemoglobin is 50% saturated. To find the P50 on the curve, first find the 50% saturation mark on the y-axis, and then drop a line to the x-axis. And this will give you the partial pressure of oxygen at which 50% of the hemoglobin is saturated. And the P50 can tell you a lot about the strength of binding between oxygen and hemoglobin in a particular tissue or under specific conditions. Let's first take a look at what the oxygen-hemoglobin dissociation curve looks like under normal conditions, and then we'll talk about how this curve shifts under different conditions. Hemoglobin approaches saturation at a partial pressure of about 80. And in the alveoli of the lungs, the partial pressure of oxygen is typically about 104. So there's plenty of oxygen, and the hemoglobin is essentially saturated. As blood passes through the systemic capillaries, the partial pressure of oxygen drops to about 40. And at this partial pressure, the hemoglobin typically unloads about 22% of its oxygen. Now the normal P50 of hemoglobin is about 27, and this assures that in normal physiologic conditions, there's still a good amount of hemoglobin that's carrying oxygen, so you won't run out of oxygen in the tissues. So what you see here is a normal physiologic oxygen hemoglobin dissociation curve. But hemoglobin doesn't unload the same amount of oxygen in all tissues under all circumstances. So in the next slide, we'll begin to talk about the factors that can influence the binding of oxygen to hemoglobin and therefore will shift the dissociation curve. So here's the 50% hemoglobin saturation mark. And for the normal blue curve, we can drop that down and find the P50 on the x-axis. And for the curve that's shifted to the right, here's the P50. And you can see in this right-shifted curve that the P50 is higher. And that means that more oxygen is required to reach the 50% saturation point of hemoglobin. So if more oxygen is needed, that means that there's a lower affinity between oxygen and hemoglobin. So a right-shifted curve is indicative of decreased affinity between hemoglobin and oxygen. So in a curve that's shifted to the right, the P50 is higher and the affinity of hemoglobin for oxygen is lower. And if the affinity is lower, that means that hemoglobin will more easily release oxygen to the tissues. So here's a study tip for you. R is for right and R is for release, because a right-shifted curve occurs in conditions that favor the release of oxygen. So think about under what conditions would it be beneficial for hemoglobin to release more oxygen. The simple answer is this: when the tissues need more oxygen. One example would be during rigorous exercise. So during exercise, skeletal muscle cells will need more energy and therefore will have increased metabolic needs and require more oxygen. And in this case, it's certainly going to be beneficial for the hemoglobin to have lower affinity for oxygen so that it can release more oxygen to the muscle cells. In general, increased metabolism in a tissue will require more oxygen and therefore will create conditions that result in a right shift of the dissociation curve. This table shows examples of various factors that will decrease the affinity of hemoglobin for oxygen and therefore will cause a right shift in the dissociation curve. And all of these factors can be a result of increased metabolism in a tissue. Cellular metabolic activity often produces acids, and acids are going to increase the hydrogen ion concentration, which is the same thing as saying it lowers the pH. Cellular respiration often gives off carbon dioxide as a waste product, and much of the energy generated from catabolic processes is released as heat, and that's going to increase the temperature of the tissue. Now for number 4, remember that human red blood cells don't have mitochondria, so they meet their energy needs solely through anaerobic pathways. And one of those pathways is glycolysis, and one of the intermediate products of glycolysis is 1,3-diphosphoglycerate, or DPG. And 1,3-DPG can be converted to 2,3-DPG. Or we sometimes see this as 2,3-BPG, or biphosphoglycerate. And this molecule, DPG or BPG, interacts with the beta chains of deoxygenated hemoglobin and decreases their affinity for oxygen. And this enhances the release of oxygen from the red blood cells. So increased metabolism in a tissue can lead to decreased pH, increased partial pressure of carbon dioxide, increased temperature, and increased DPG. And let's see how these factors affect our oxygen hemoglobin dissociation curve. First, let's remember that a right shift in the curve is induced by conditions that favor the release of oxygen. And you can see on a right-shifted curve that you have an increase in P50, and this means a decrease in the affinity of hemoglobin for oxygen. And we haven't yet talked about a left shift in the curve, but by the same logic, a left shift of the curve occurs under conditions that favor the loading of oxygen onto hemoglobin. So L for left and L for loading. And a left shifted curve is going to have a decrease P50, which means an increase in affinity of hemoglobin for oxygen. So in the left shifted curve, the hemoglobin is going to want to grab onto oxygen and load more oxygen onto the hemoglobin. So while increased DPG will lead to a right shift in the curve, decreased DPG will lead to a left shift. And for temperature, an increased temperature leads to a right shift, decrease in temperature to a left shift. High acidity, which is low pH, leads to a right shift, and low acidity, which is high pH, leads to a left shift. And one study tip that we use to remember the effects of the various factors on the dissociation curve is this acronym, cadet face right, where C equals CO2, A equals acidity, D equals DPG, E equals exercise, and T equals temperature. And you can use this acronym to remember the factors that are going to cause a right shift in the dissociation curve. And the last thing we're going to do in this tutorial is to make some comparisons between human adult hemoglobin and some other oxygen-binding pigments. And first we'll take a look at fetal hemoglobin. In humans, fetal hemoglobin is also composed of 4 polypeptide globin subunits. The alpha globin chains are the same as in adult hemoglobin, but in the fetus, the 2 alpha chains are combined with 2 subunits of gamma globin. And these gamma globin chains persist for about 6 to 12 months after birth, as they're eventually replaced by the adult beta globin chains. Now, fetal hemoglobin has a stronger affinity for oxygen than adult hemoglobin, and this is physiologically important because it allows the fetal blood to efficiently extract oxygen from the maternal blood. And I see that's missing on this slide, so we'll add it in. Fetal hemoglobin extracts oxygen from the maternal blood. So before we leave this topic, I'll pose this question to you. What would the oxygen hemoglobin dissociation curve look like for fetal hemoglobin? And to answer this question, it will help to draw the comparative curves between adult hemoglobin and fetal hemoglobin. You should also be familiar with myoglobin. Myoglobin is the oxygen-binding protein that's present in the cytoplasm of muscle cells. And you can see in this figure that myoglobin is composed of a single polypeptide chain and a single heme group. And here's myoglobin, and on the right is hemoglobin. And you can see in this figure that myoglobin is composed of a single polypeptide chain, and it has only a single heme group, so it can only bind a single oxygen molecule. So take just a moment to think about how the oxygen myoglobin dissociation curve would be different from the oxygen hemoglobin dissociation curves that we've looked at so far. The red line on this graph illustrates the binding between oxygen and myoglobin. Now remember that the sigmoidal shape of the oxygen hemoglobin curve, which we see here in blue, is due to the fact that the oxygen binds cooperatively to the 4 subunits of hemoglobin. Myoglobin only has one binding site for oxygen, and therefore it doesn't exhibit cooperative binding, and the curve shape is what we call hyperbolic. So that's this curve here shown in red. So you should note the differences in the shapes between these two curves. So I have one last question for you in this tutorial. Which do you think has a higher affinity for oxygen, hemoglobin or myoglobin? And why would it be important physiologically for these two molecules to have a different affinity for oxygen? Think about the physiologic interactions between hemoglobin and myoglobin. This completes the tutorial for the oxygen-hemoglobin dissociation curve. If you have any questions about this tutorial, please feel free to ask your lab instructor during the lab or to contact us through the discussion board.